Hello, my name is Grant Kramer, and I am Professor Emeritus at the University of Nevada, Reno. Today we will be continuing my series on mineral nutrition of plants. This will be the second in a series of videos.
Mineral Nutrition of Plants, Part 2, the Grapevine Specifics. Here are some things to consider for grapevine-specific nutrition. First of all, grapevines grow like weeds. Most fertile soils may provide an adequate mineral nutrition for the vines, and fertilization may not be necessary or may even be a waste of money. Are your grapevines growing and producing adequately? Did you do a soil analysis and tissue analysis for mineral nutrient to determine if fertilization is needed? Does your vine show any mineral deficiency or toxicity symptoms? And I add a cautionary note here. Some symptoms that you can observe may be confused with disease symptoms. So just because it looks like a mineral nutrient deficiency may not mean that it actually is. Mineral nutrients can also impact the quality of your wine, good and bad. So how to test for adequate mineral nutrition? First, look at the vine to see if there are any symptoms. Second, perform a soil analysis. Soils are usually collected from different locations and at different depths to get a good average survey of your soil profile. Perform a tissue analysis. It is recommended to analyze the petioles of the leaves opposite to a cluster at the flowering stage. These have been found to be the most reliable and sensitive tissue analyses. Consult with your soil and mineral analysis company for specific details. The mineral nutrients to consider in grapevine are nitrogen, phosphorus, potassium, and magnesium, all of which are macronutrients. And the micronutrients to consider in grapevine are iron, manganese, zinc, boron, and copper. There are other essential mineral nutrients for plants, which I have discussed in part 1 of this series, but these are the ones that are most important for grapevine. Nutrient deficiency symptoms are hard to find in grapevines, but there are a number of sites that have shown nutrient deficiencies that have been observed or tested for, and I've listed them here. I personally have not been able to test grapevines for nutrient deficiency, so we are relying on observations made by others, and those vary with the cultivar that you're looking at. So in these websites, you may find different visual symptoms simply because of the difference in cultivar or the difference in the concentration of the deficiency. But I recommend looking at all of these as they are very useful and very interesting. So as I mentioned down here at the bottom of the slide, nutrient deficiencies vary with the grapevine cultivar. So it's important to verify your nutrient deficiency symptom with either a tissue and or a soil analysis to confirm that nutrient deficiency. So let's start with nitrogen. Nitrogen is required in the largest amount in the vine. It is a constituent of proteins and chlorophyll. The deficiency symptoms for nitrogen result in stunted growth, reduced vigor, reduced leaf size, and shoots. Nutrient deficiency symptoms for nitrogen start with yellow leaves, which is due to a loss of the chlorophyll molecules. This starts in the early stages of the deficiency in the older leaves first, because nitrogen is a mobile element and is recycled in the plant to the younger tissues. And if you'd like to know more about that, see part 1 of this series, where I give a more complete explanation and reasoning for this. But with additional deficiency or a greater deficiency, this yellowing will spread throughout the entire plant. Managing nitrogen in grapevines is very important for vine balance and wine quality. That is, the optimum nitrogen needed for optimum vine growth is not equal to the optimum wine quality. In fact, if you provide nitrogen to provide optimum growth, it will be too much and it will impact negatively your wine quality. So too much nitrogen can cause excess vigor in your vine, resulting in crowded canopies, which can lead to greater amounts of mildew, harder or greater difficulties in getting your spray into the canopy. It also reduces fruit set and bud fertility. And as I mentioned earlier, it results in reduced wine quality. So I love these figures that I'm going to show from Rogers et al. 2021 from Australia, where they've done a very nice job of visually looking at the mineral nutrient deficiencies in grapevine. And in this slide shows an excellent image of the changes in, or the progression of, nitrogen deficiency in Shiraz, with less deficiency in the left-hand side and more severe deficiency on the right-hand side. So the symptoms are a uniform loss of greenness and a yellowing across the entire leaf blade. The mature leaves are usually more yellow than the younger leaves. And again, this is because nitrogen is a mobile element in the plant. So here are some important nitrogen concentrations to consider. The recommended petiolar nitrogen at bloom should be greater than 350 parts per million nitrate nitrogen and less than 1,000 parts per million. Plant vigor is another way of looking at your nitrogen use or your nitrogen concentration in the vine, as it is directly proportional to the amount of nitrogen and water that you apply to the plant. So too vigorous a plant is probably a good indicator that you've added too much nitrogen. Slower growth might be as a result of some other nutrient besides nitrogen, so one must be a little bit careful about that as an indicator. Leaf blade measurements can also be made, but they're not as sensitive as leaf petiole measurements, and therefore it's recommended the leaf petiole measurements at bloom are the ones that should be used. Another way of measuring nitrogen, that is a good way to measure nitrogen at the end of the season, is the yeast assimilable nitrogen, or YAN. You measure this in the grapes and or the grape must, which is grape juice. Less than 140 milligrams of nitrogen per liter of must is considered deficient, and 180 milligrams of nitrogen per liter is considered adequate or sufficient. Low must YAN leads to low yeast populations and poor fermentation vigor. Increased risk of sluggish or stuck fermentations, increased production of undesirable thiols, those are sulfur compounds, and higher alcohols. Lower production of favorable fruity esters and long-chain volatile fatty acids. High must YAN leads to increased concentrations of ethyl acetate, acetic acid, and volatile acidity. Haze-causing proteins, urea, ethyl carbamate, and biogenic amines. Ethyl acetate concentrations are proportional to the amount of YAN. That is, the greater the YAN is equal to greater the ethyl acetate in your wine. So let's talk a little bit about nitrogen applications. Grape wine quality compounds will benefit when nitrogen is added to the vine to alleviate nitrogen deficiency in the grapevine. Nitrogen is also beneficial if added in small amounts when vine nitrogen status is adequate to balance the loss of the nitrogen after grape harvest, because you're removing nitrogen with the grapes, and help to maintain a nitrogen balance in the vine over the seasons. Do not add nitrogen to vines when the nitrogen status is high, or grape must and wine quality will be reduced. A general recommendation for first year's planting is to add 10 pounds of nitrogen per acre, and then thereafter add as needed based upon the amount of crop removal. To get an idea of how much nitrogen you're removing, it's been shown that 12 tons of fruit per acre equals to about 50 kilograms of nitrogen per hectare, or 44 pounds per acre. Phosphorus is a major macronutrient. It is an important component of membranes and energy molecules. Phosphorus deficiency is rare. Phosphorus deficiency shows up when petiolar concentrations are less than 0.2% at the flowering stage, or bloom. Deficiency symptoms result in stunted growth, smaller, darker leaves, and in some reports, purplish or bronzing of the leaves occurred. But in other reports, no leaf symptoms were observed. In addition, phosphorus deficiency results in reduced fruit set and yields. Potassium is an important macronutrient. It is an important soluble osmotic component that maintains charge neutrality, water uptake, and turgor in the living cells. It is an important component of grape juice. Increased uptake of potassium results in higher pH in the juice, which can have problems with wine quality if the pH becomes too high. Higher concentrations in the plant may give the vine the ability to be more drought tolerant. The vine is considered potassium deficient if petiolar potassium concentrations are 1% or below at bloom. Potassium concentrations are considered excessive if petiolar concentrations are 3.25% or higher. This is a nice slide of the visual symptoms for potassium deficiency, again showing a progression from left to right of more moderate to more severe potassium deficiency. And in the upper section, you see what it looks like for Chardonnay, and in the lower image, you see what it looks like for Shiraz or Syrah. So in the beginning, symptoms first appear on the left-hand side along the edges of the margin, and you can see a slightly reduced coloring and the beginning of the formation of brown spots along the margin. This is more apparent in the Chardonnay leaf sample than it is in the Shiraz sample. And then as the potassium deficiency progresses, it begins to accumulate more browning along the margins so that the browning spots can begin to coalesce. And eventually it progresses to consume the leaf, as seen here on the right-hand side. Magnesium is a macronutrient required in the plant in large amounts. Magnesium serves many roles in the plant, but one of its most important functions is that it is a component of the chlorophyll molecule. So magnesium deficiencies show up as interveinal chlorosis of the older leaves, and this is important in order to distinguish it from iron deficiency, which also shows interveinal chlorosis, but in the younger leaves. And again, if you see my first video in part 1, this is due to the mobility of the ions. Magnesium is a more mobile element and can be recycled to the younger leaves, whereas iron cannot. Some soils in the western USA, and there are not many of them, but there are some present, that have too high of magnesium in them. And this can cause problems and resulting in magnesium toxicity. And one way to counter this is to supplement the soils with calcium sulfate. Petiolar magnesium concentrations at bloom should be above 0.2 to 0.3% to be sufficient. And again, just to give examples of the differences one might see between magnesium and iron deficiencies, we can look at the progressions of magnesium deficiency in Shiraz in the upper figure, and we can see the progression of iron deficiency in Shiraz in the lower images from left to right. Again, the iron deficiency is going to show up first in young leaves, whereas the magnesium deficiency is going to show up in older leaves. You can see differences in the pattern. In fact, it's mentioned here that the interveinal chlorosis and iron deficiency shows more finer veins, and it is quite apparent in this figure. But I'm going to tell you this varies with cultivars, and I'm going to show you iron deficiency in Pinot Noir that looks more like the magnesium deficiency you see here. So the best way to differentiate between magnesium and iron deficiency is where they occur in the plant. Does it occur in the older leaves first, and then progress to younger leaves? Or does it appear first in younger leaves and progress to older leaves? If it starts in older leaves, then it is magnesium deficiency. If it is starting in younger leaves, it is iron deficiency. Iron is a micronutrient, and it's required in plants in small amounts. Iron is important for chlorophyll formation and acts in numerous electron transport functions, especially in the photosynthetic electron transport chain. Iron deficiency symptoms are intraveinal chlorosis of the younger leaves, as I just explained in detail. Soils above pH of approximately 7.5 will have low available iron, and therefore plants will suffer from iron deficiency as a result. The best way, or the preferred way, to correct for this is to lower the pH of your soil. Alternatively, you can spray your vines, the leaves, with an iron chelate foliar spray, and this will alleviate the problem. Petiolar iron concentrations at bloom should be above 75 parts per million to be sufficient. As I mentioned, iron deficiency can look a bit like that magnesium deficiency. So here is a picture of high soil pH-induced iron deficiency in a young Pinot Noir vine in my backyard. And to confirm that it was indeed iron deficiency and not, say, magnesium deficiency, I sprayed the leaves with an iron EDDHA chelate foliar spray, and this alleviated the symptoms. That is, the leaf turned more green and no longer had intraveinal chlorosis. And this is proof that the plant was deficient in iron and not some other nutrient like magnesium. Here are a couple of other images of iron deficiency in the field. The one on the left is showing light yellowing of the growing tips. If you look carefully at the bottom of the image on the left, or on plants up at the top end of the plant, you can see lighter yellowing, but not intraveinal chlorosis at this point. So this is in the early stages of iron deficiency. On the other hand, on the right, you can see a severe iron deficiency where the entire vine is looking yellow. And you can't really see the progression from the top to the bottom very easily. This is severe, and in fact, you don't see much in the way of green veins. Almost the leaf is completely turned yellow. Manganese is a micronutrient, and it is needed in small amounts in the plant as well. It is involved in the synthesis of chlorophyll and nitrogen metabolism. It is involved in O2 production at the beginning of the photosynthetic electron transport chain. According to Australian researchers, manganese deficiency symptoms are often similar to magnesium and zinc deficiency symptoms, showing up as intraveinal chlorosis of the older leaves. High pH soils can limit the availability of manganese to the vine, and petiolar concentrations of manganese above 20 parts per million at bloom are considered sufficient or adequate. Zinc is a micronutrient that is needed in small amounts in the vine as well. Zinc serves a variety of roles in various metabolic processes. Zinc deficiency symptoms show up in the fruit cluster at first, in mild deficiency symptoms, and when it's more severe, it can also be seen in the leaves. The clusters have shot or hen and chicken berries. That is something like what you see on the right, where there are large berries, and then there are small berries that don't really develop. Leaf symptoms are described as a mottled look between the veins and stunted growth. Again, soils high in pH or in sandy soils with low nutrition can have reduced zinc availability. Zinc foliar sprays can alleviate these problems and are especially useful just before bloom. Petiolar zinc concentrations at bloom above 20 parts per million are considered sufficient. Boron is a micronutrient that is needed in very small amounts by the vine. Boron appears to be involved in cell wall structure and cell elongation functions. Severe boron deficiency will show up as dieback of the growing shoot apices. Milder boron deficiency can result in poor fruit set and seedless berries. Boron toxicity is another problem which can show up first at the leaf margins as cupping and later with brown spots along the margins. Soils with boron deficiency are not that common, but toxic concentrations commonly show up in alkaline soils with low rainfall. Boron is very mobile in the soil, and it can be leached with high-quality water. Boron deficiency is overcome with foliar sprays. Petiolar boron concentrations at bloom that are less than 25 parts per million are considered deficient, and greater than 125 parts per million are considered excessive. So there's a very narrow range in which boron is sufficient or adequate. This is a nice image, again from Rogers et al., on the progression of boron toxicity in Shiraz from left to right. So in the beginning, again, you start to see some brown spotting along the margins. You could interpret that maybe as similar to potassium deficiency, but if you compared it to the image with potassium deficiency before, you would see some significant differences. The spots here are smaller. And then you see as it progresses that it continues to remain as smaller spots that don't coalesce as much. And eventually you can get cupping in the leaves and a reddening or continued development of these lesions. Copper is a micronutrient that's needed in very small amounts in grapevines. It is a component of a number of enzymes, including some involved in photosynthesis. Copper deficiency is rare. Copper toxicity is more common because it is a component of the Bordeaux mixture fungicide that's used to treat mildew problems in wet regions, in particular for downy mildew. Copper accumulations in the soil have occurred because of the repeated spraying of these copper-based fungicides, which then allow for the accumulation of copper in the soil. Copper transport to the shoot is very limited. Consequently, copper toxicity is rarely seen in the shoot or the leaves as symptoms of copper toxicity. Copper toxicity, in fact, occurs more commonly below ground by inhibiting root growth. So this is not something that one can observe easily. Petiolar concentrations at bloom below 5 parts per million are considered low or deficient, and copper concentrations above 25 parts per million are considered excessive. So in summary, soils may have mineral nutrient deficiencies or toxicities. It is important to conduct mineral nutrition analyses before fertilizing your grapevines. Otherwise, you may be giving them excess nutrients, which may have detrimental effects on the vines, and or be wasting your money by putting on something the plant doesn't need. Mineral nutrients are monitored by visual symptoms, soil, and tissue analyses. Soil analyses are not always a good indicator of grapevine nutrition. Petiolar analyses are better indicators of that vine's nutrition. Nitrogen is the most important mineral nutrient, and careful management of it is needed to provide balanced growth and optimum fruit quality. Iron deficiency is the most common nutrient deficiency found in soils with high pH. Understanding the mobility of the ions in the plant, as I described in part 1 of this series, can aid you in the identification of the nutrient deficiency symptom that you are observing.
Well, that's it. I hope you enjoyed this video. If you liked this video, then please like it on my YouTube channel. And subscribe to my channel if you would like to hear additional videos like this one. Have a great day.